and ecological genetics and genomics of forest trees, and investigates the capacity of populations to adapt genetically to projected future climates. Dr. Aitken is the director of the Center for Forest Conservation Genetics, which generates and disseminates knowledge important for the genetic conservation of forest trees. And she's also the leader of the Co-Adapt Tree Project, which provides recommendations and delivers genomic tools and strategies to forest managers on assisted species migration for several important tree species in North America. Dr. Aitken has won several awards for her excellence in teaching and being an elected fellow of the Royal Society of Canada in 2017, and being awarded the Genome BC Award for Scientific Excellence. She does, I'm echoing. She does a lot of interesting and important work on how to make our forests more resistant and resilient to climate change. And myself and Katie Nigro, who are kind of co-hosting her visit and who wrote this speech, are personally really inspired by her ability to bridge the gap between science and management and provide solutions and tools to help forest managers address the many challenges that they are facing today. So without further ado, please help me welcome Dr. Aitken. Ooh.
Thanks very much, Miranda. I'll just get my presentation up. That was a really nice introduction. And I hope everyone can hear me okay. I'm just going to arrange my screen so that I can see some of you and speak to you at the same time. So I really wish I was there in person with you. It's complicated times. And well, the border, officially, it's still a complicated matter going back and forth with the testing requirements and things, but I really wish I was there instead of here. So without further ado, I'm going to talk about conifers, as Miranda suggested, and talk about climate change. We have had a crazy year in British Columbia. I've been studying climate change and forests since the early 1990s, and it was always something in the future, projecting impacts in the future. Well, this year we had temperatures, record-breaking temperatures in June with the heat bomb, a village that experienced the highest temperature ever in Canada by several degrees. It was 49.6 degrees Celsius one day, and the next day that village of Lytton burned to the ground. So this was a real wake up, but that wasn't the end of it. We had an extended drought. And the trees were suffering greatly from the combined effects of the drought and the heat. And we had a state of emergency declared. 2 weeks ago, a tornado tore through the forest next to you and arrive. And it was forecast to bring a lot of rain, but it brought a lot more than was expected. And between Saturday and Monday, up to 11.5 inches of rain fell on the lower mainland. This has resulted in Vancouver being cut off from the rest of Canada. Every major highway is cut off. There's no way to drive now from Vancouver to anywhere else within Canada. The only place we can go is Seattle, and a state of emergency was declared about an hour ago. Extensive flooding, huge infrastructure damage, and there's a lot of people who are stuck far from home because it was a long weekend here. And this is bad, but it's the tip of the iceberg. I like this figure that came out just before COP 26. Showing the different trajectories. And wouldn't it be nice if we were on this below 1.5 pathway with these colors projecting future temperatures? Pledged policies, we're on this trajectory, and you can see it's going to get a lot warmer and a lot weirder. current policies before COP, this was before the pledges at COP, are taking us into much higher levels of warming than that. So back to conifers. So I study mostly, and my group studies the adaptation of conifers to climate, and the odd angiosperm thrown in. And the reason we study conifer populations and climate is that we see fairly strong local adaptation to climate in our widespread species. This is an example from Sitka spruce. Here above each of is the provenance of these are all the same age, cut out of a common garden experiment, and they all represent the average saw for each of those provenances, and we see a steep climb. The trees from California, they grow big because they grow for a long time. They have a long growing season. And at the other end of the spectrum, we've got trees from Alaska that are adapted to a very short growing season and achieve quite deep cold hardiness compared with the trees from California. And this is a pattern we see for a lot of our species. The only species we've studied that we don't see in a benign environment, that we don't see this pattern in, actually is Gary oak, which is a maritime climate native of the West Coast. So thinking about how populations of trees or anything else can respond to climate change, we have a few options. First of all, we have a shifting climatic niche, which is schematically indicated by this dashed line. And we've got some natural species range. And so we might have natural migration. We expect to have natural migration from the leading edge into areas that have become favorable. We might have adaptation over generations from standing variation and from gene flow. And I know Ruth Hufbauer's group works on this process, which is evolutionary taking edge of migration, and how that process will unfold is, well, we will see, but we're already seeing some of that. But for trees, these are all slow processes in a rapidly changing climate. And just to give you some sense of the conifers we work on, I did a virtual visit to Michigan State a few weeks ago, and if you think about E. coli in the long-term evolutionary experiment of Richard Lenski's, he's achieved over 75,000 generations in his career, and that's a 5 megabase genome. Arabidopsis, well, maybe a researcher could complete 150 generations in their career. And the genome size is about 135 megabases. Well, we work on conifers, and a tree breeder would be lucky to turn around 2 generations in their lifetime. It's more likely 1 generation. And we have a 20 gigabase genome, so about 7 times human. So it's challenging. And this is a tree called Big Lonely Doug, by the way. It's one of the biggest Douglas firs in Canada, and it's standing in the middle of a clear cut, tragically. So what about migration? Well, Joanne Elouette was a PhD student in our group, and she studied the colonization of Sitka spruce in Alaska from the mainland here, including Seward, over to Kodiak Island. And so Sitka spruce only reached Kodiak Island about 500 years ago. And tree cores with genetic samples to recreate the dynamics of colonization on the island. And so a huge tree that was probably an early colonizer arrived, had few trees or no trees around it. You can see by its structure. And around this, we find closed canopy forest. And then out on the edge of the Sitka spruce colonization, we see trees that are spotted out across this previously treeless environment. And so by combining the tree cores with the genetic data, Joanne was able to look at both the demographics of the stands that she sampled, and the genetic diversity in those stands. And so if we look here, the open white bars represent an old forest at Seward where Sitka spruce has been for a couple of thousand years. And this is on the x-axis, we have the date of establishment. So this shows you the range of ages in a forest that hasn't had a major disturbance for a while. And that compares with, on Kodiak Island, the gray bars. And so Sitka arrived back here in the 1500s, and it slowly increased in numbers until about 1700, when the population really took off. And this corresponds to an increase in genetic diversity that would have arrived via gene flow, via pollen. So we see this slow lag in genetic diversity and in demographics. So this process of trees shifting range is a very slow one, in part because trees take up to 20 years to start reproducing, and because they have limited seed dispersal, etc. In response to the end of the last glacial maximum. So what we think about a lot in my group is the process of assisted gene flow, which is shifting seed sources for reforestation or restoration to accelerate this process of adaptation to new climatic conditions. You also might think about assisted species migration. There's a little bit of that going on in British Columbia, whereas assisted gene flow is now in policy for reforestation. And of course, we expect to lose populations at this rear edge. And so we need to be doing ex situ conservation and a lot of monitoring. And assisted gene flow, I just want to define here, is the intentional translocation of individuals within a species range to facilitate adaptation to anticipated local conditions. And this is related to, but not the same as, genetic rescue, which has worked on. And the idea is that you are providing both a release from inbreeding and an infusion of genetic diversity to rescue populations, but that's not necessarily intentional with respect to adaptation to new conditions, but certainly they're very related concepts. So in my group, we combine genomic data with climatic data and phenotypic data to understand adaptation to climate. And the things I'm going to talk about today are, first of all, what are the general phenotypic and genomic patterns we see associated with local adaptation to climate? I'll just mention very briefly whether we see convergent evolution among conifer species. I'm going to talk about a little bit about the effects of selection for faster growth on climate adaptation, looking at the effects of tree breeding on many different populations. And then a question that I talked to Jeff Morris quite a bit about earlier today is, do phenotypes and genomic data tell us that populations are locally adapted to the same climatic drivers? And I think this is a really important point, because in some cases we will only have genomic data to evaluate what is driving local adaptation. In species we can't do common gardens, so we need to answer that. And then I'll touch on some recent results looking at genomic offsets as from genomic data as a predictor of the extent of future population maladaptation. And most of this work is going to come from the two major genomic projects we've had. The first was called Adaptree, the second was called Co-Adaptree. These are large-scale applied genomics projects that have been funded by Genome Canada and other regional genome research centers. And these projects target widespread, economically and ecologically important species. So I won't be talking about things like white bark pine, which we've also done some work on because we have the genomic data. We were able to get genomic data for these species of widespread importance in forestry. So first set of questions here. So with Adaptree, we focused on lodgepole pine, which I know you have lots of in Colorado also, and something we call the interior spruce complex, which includes white spruce, a boreal species, Engelmann spruce, which you have at high elevations in Colorado, and their hybrid populations. And in British Columbia, most of our interior spruce populations are heavily intergrassed between these two species, and sort of many generations of hybridization have gone on. And these are managed as a single species, this complex. So the map on the left here shows you some of the phenotypic results from our work with lodgepole pine. So here we have grown 281 provenances in growth chamber environments, and we've done artificial freeze tests on them in the fall. And so what this map shows you is the location of those seed collections. And if the circles are dark blue, they were very cold hardy. And if the circles are red, they were very cold susceptible. They had high levels of injury. And you see a pattern that goes from north to south in the level of cold injury. And that corresponds to, not surprisingly, the occurrence of low winter temperatures. So the map on the right shows 30-year extreme minimum temperature, but I could show a similar map for mean coldest month temperature, or the difference in temperature between summer, the warmest month, and the coldest month, otherwise known as continentality. And these were the results for lodgepole pine. But if we look at interior spruce, we see a very similar pattern. Again, we've got this pattern that corresponds, not surprisingly, with low temperatures. Over all the species we've studied, the dominant signal is adaptation to low temperatures. And the phenotypic trait that gives us the strongest differentiation among populations is cold tolerance. What these two-dimensional maps don't show you is also the pattern of variation with elevation that is climate related. And Sam Yeaman and Kay Hodgins, as part of this project, looked at the extent to which there was convergence between spruce and pine in terms of the genes underlying this adaptation to low temperatures. So we had done exome capture of about 23,000 genes in each of these species. And they looked at, first of all, both genotype environment associations and genotype phenotype, or GWAS associations, and identified genes that were associated with the adaptation to low temperatures. And then what we did was took the top candidate genes in each of these species just based on raw row scores, so raw correlations, no population structure adjustment. And they looked to see what the overlap was. And in fact, we found that there were 47 genes that were in common between the two species. And this is interesting because these genera are separated by about 140 million years of independent evolution. So Sam Yeaman's group now at the University of Calgary is running these tests on additional species as part of the Adaptree project, and looking at about 8 conifer species in North America and in Europe to look for convergence. This method of looking at convergence using raw correlations is one that we found to be effective, because when we adjust for population structure, for neutral population structure with the species we're working with, we generally strip away also signals of local adaptation because the recolonization patterns of these species are along climatic gradients. And so the demographic structure of these populations is collinear with some of the climatic gradients. So this cross-species comparison has been interesting. Now, you might say climate's warming. Why do we care about cold? And we've done a lot of work on cold hardiness. Well, if we think about assisted migration within range or outside of range, as we move trees to colder environments, we are going to increase the risk of cold injury in the short term. So we need to understand the effects of those transfers. And we also see that strong local adaptation for low temperatures. This is a paper that just came out that's looking at the risks of transfers in different directions. This work that was led by Jihao Han Sang, PhD student of Andreas Hammonds at the University of Alberta, very nicely showed that latitudinal transfers carry less risk of cold injury than elevational transfers. Okay, I want to switch gears a little bit and talk about what the effects of selection for faster growth are on climate adaptation. So we know at a population level, as I showed with that first image of Sitka spruce, that generally populations from milder environments are less cold hardy and faster growing. Populations from colder environments are slower growing, but more cold hardy. So when we select within populations for faster growth, are we altering the climate adaptation of those populations? So this was work, this was part of the PhD work of Ian McLaughlin, published earlier this year. And so what Ian did was each of these colored polygons represents one breeding zone, one tree breeding zone for lodgepole pine in British Columbia on the left and Alberta on the right. And each dot is a natural population collection from within that breeding zone. So this was the comparison between natural and selectively bred trees. Most of the breeding had gone on for 2 generations in BC, 1 generation in Alberta. And so what Ian did was we have a 2,000 SNPs on it that genotyped well. And and it had 18,600 contigs, so gene models, transcriptional contigs. So first of all, Ian looked for the 1% of candidate SNPs that had the lowest p-values in GWAS for each of 4 seedling traits. So that's 1 SNP per gene. He took the SNP that was most associated with height growth, which was the trait that was under selection. So 1% of 18,600 contigs, you end up with 186 SNPs per trait. And then for each SNP, 1 allele was labeled the positive effect allele. That was the allele associated with an increase in trait value. So for height, it would be the allele that was associated with greater height growth. And he looked at 4 seedling traits: height, the timing of growth initiation in the spring, the timing of growth cessation in the summer, and cold injury in artificial freeze tests. These are highly polygenic traits. So here, the x-axis shows you the average frequency of positive effect alleles of those 186 alleles for each trait. And this shows you the distribution of those average allele frequencies. The red line is for the natural populations. The blue line is for the selected populations. And so here we have height and cold injury in 2 different regions. So this is broken down just into 2 broad regions: Southern British Columbia, which is warmer, and Central British Columbia, which is colder. And what you can see is the breeding has just shifted the frequency of many alleles just a little bit. So we're seeing small effects across many loci here. And generally, these effects look pretty similar between the warmer and colder breeding zone for height and for cold injury. But when we look at the 2 phenology traits, we see quite a different response. So first of all, I need to explain this. This is this is a plot showing the change in allele frequency with selection. So each line is one of those 186 SNPs. And the red SNPs happen to be SNPs that were associated with all 4 traits. The blue SNPs are those associated with height, which was the trait that was selected on. And you can see that most of these SNPs that were associated with height in a GWAS increase in frequency. Makes sense. You're selecting for a greater height growth. And we see increases for the most strongly associated of a little over 10% for height. But we also see increases in the frequency of cold injury associated SNPs, many of which overlap with the height SNPs. And the warmer region and the colder region, we see pretty similar patterns, not quite as big of increases in allele frequencies for many of these SNPs, but pretty similar. But when we look at the phenology traits, we see big shifts in almost all of the associated SNPs for both growth initiation and growth cessation in the warmer region, but in the colder region, we don't see nearly as much change. Now, these trees are being selected based on common garden experiments that are out in these regions experiencing these temperatures. And so it looks like selection for faster growth in the warmer region is really reflecting kind of a release of climate on those growth rates. But in the colder, and I think this just shows that phenology, growth, and cold injury are highly polygenic and very much interrelated in these trees. So that's a lot about cold and about phenology. We see local adaptation for those. We've known that for a long time. Foresters have known that these things vary for a couple of centuries. So this isn't new knowledge. But right now, we're pretty interested in drought and drought tolerance. And I think those of you in Colorado probably are as well. And of course, drought mortality is really increasing across Western North America. And we're seeing also increases in insects that are able to really explode on stressed trees and in warmer climates, like the big bark beetle epidemics. And but just thinking about heat and drought and temperatures, and then we had an extended drought in BC, and we've seen quite a bit of dieback on different tree species. And here we have Douglas fir. And so on the coast, we saw a lot of needle loss on Douglas fir, and it happened to be often associated with a needle pathogen. So it appears this is really a needle fungal epiphyte, but it goes into stomata, and it appears that effect of blocking stomata from fully closing may have exacerbated the effects of either the heat or drought. Can't separate those. In the interior variety of Douglas fir, we see a lot of top dieback from the drought. So we're seeing phenotypic effects and younger trees that are dead, in fact. Rafael Ribeiro is a PhD candidate, and he has been studying drought hardiness in Douglas fir as part of CoAdaptry. And so we've been working with 80, and Brandon Lind is a postdoc who's been doing much of the bioinformatic work for this study. So we have 87 populations of Douglas fir. They include both the coastal variety Menziesii and the interior variety Glauca, which are separated by a couple of million years evolution, although there's certainly gene flow between them. And we did pooled exome sequence capture on these population exome capture. And then we have common garden experiments for phenotyping drought as well as cold. So this is one of Rafael's drought experiments, and you can see it's a split plot design. All 87 populations are in here, and we've got drought applied in a greenhouse from over 150-day period. And here you can see very clearly the control and drought treatments. Rafael used chlorophyll fluorescence to monitor the decline in the health of the photosystem of these trees. And so you can see the wet treatment had no decline, but the dry treatment, the chlorophyll fluorescence indicates that descent towards death, basically, of these plants. Now, the purple lines are interior Douglas fir, and the green lines are coastal Douglas fir. And the interior Douglas fir was considerably more drought hardy than the coastal Douglas fir. Not unexpected based on the environments that they come from. Some variety glauca in Colorado. But when we look within variety, so there was about 18% of the variation was due to differences between interior and coastal Douglas fir. But when we look within coastal Douglas fir, there was essentially no among population, there's variation. Interior Douglas fir, we did see some among population variation, but certainly less than we see for cold hardiness or for bud phenology. Now, using the slope of that decline in chlorophyll fluorescence as a proxy for drought hardiness or drought tolerance, if we put both varieties together, we can see a weak negative, expectedly negative relationship between the amount of precipitation or drought hardiness. But if you look at these the varieties separately, there's really no relationship within variety. And we saw some unexpected results. There were, if you look regionally, and granted there are few data points for these regional analyses, but it's interesting that here in Idaho, Western Idaho and Eastern Washington and Oregon, there's actually a positive relationship there in between precipitation and drought hardiness, whereas in California, we see the expected negative relationship. And in some cases, the relationship is with mean coldest month temperature rather than, or with temperature related variables, more so than with precipitation related variables. I think all of this reflects the fact that drought is, well, first of all, drought's very hard to phenotype. Secondly, drought varies spatially. It's very spatially heterogeneous. Within a small area, you can have big differences in soil water, which is what really matters. Is going to be very different. And it may be that that scale of variation is too fine-grained for local adaptation to occur, given the high levels of gene flow. But we still, drought's important, and we will continue to study it. So Raphael also did GWAS. He did a case-controlled GWAS on drought, where he took 20 populations, 10 from each variety, and within each population, he selected the 10 least hardy and the 10 most hardy trees after severe drought. And so doing that GWAS, he found about 1,000 SNPs that were associated with drought in each variety, but found only 2 SNPs that overlapped between the 2 varieties and only 8 transcriptional contigs that overlapped. So we're really talking about very different genetic makeup within the coastal and the interior Douglas fir. And I think that the analyses of variation also show that we have to be careful about including samples across too broad of a geographic and climatic and population structure range, that we really need to do our analysis within variety. We're continuing work on drought. Beth Roskilly is running experiments on Western larch, and we're really interested in larch. It's a deciduous conifer, but it's actually a sister genus to Douglas fir. And Judith Nuhu is looking at within population variation in drought hardiness within breeding populations of both coastal and interior Douglas fir. So one of the most important things in my mind, if we want to do any translocations of any species, because the populations are adapted to. And of course, with trees and with other plants, we can do common garden experiments and look at relationships with climate, but there's a lot of species that we just can't get that sort of data with. So for our trees, do we see the same climatic variables as being important driving local adaptation? And these were analyses done by Colin Mauney, and he's actually now the for everything to do with forests and land in British Columbia. So he took the data that we had for lodgepole pine for these 280 populations of lodgepole pine, and they were genotyped again for these 32,000 or so SNPs, and trees per population. So it was a big sample. And he looked at the degree to which we see the same climate variables as being important. So here we have a seedling common garden experiment. And so this graph looks at our ability to predict climate from genotypic data or from phenotypic data. It's explained phenotype for seedling phenotypes, and this is the ability to predict principal climate variables. So each data point here is a climate variable. You can see there's a relationship here. We see that low temperature the y-axis is climatic variance explained by principal components associated SNPs. Environmental temperature variables. So in both of these data sets, we identified that low temperature is the strongest signal in these data sets, but not in the phenotypic data, for example. But this is just seedling traits. And if we look at a long-term provenance trial, which is a field common garden experiment on many sites, a partial reciprocal transplant experiment, we see a much stronger relationship here. And so again, low temperature variables show up strongly in both data sets. We do see a bit more signal of moisture in the genomic data than in the phenotypic data. And this may reflect periodic episodic extreme events that we don't sample in our field. Working with our tree species, this tells me that we can get a pretty good signal of climatic drivers that are important from just the genomic data. So one of the things that we want to be able to do for the future is to be able to predict the maladaptation of populations in the future and vulnerability to future climates. And so we can use various types of data for this. We can use phenotypic data. We can use genetic or genomic data. We could just use climate. Or we could use geographic variables, which is kind of the old way we used to use geographic variables exclusively to control seed movement because they were proxies for climate. And so if we want to use phenotypes, we can this is a very simple depiction of something we could do. Here are a couple of genetic clines for traits in Sitka spruce. We've got mean and height growth. And for some focal population like this triangle here, we can look at the current phenotype, and then we can look at predicted warming and look at what phenotype is currently associated with environments that warm. And here we call this adaptational lag, but that would be like a phenotypic offset. Very simple. And this is assuming that local populations are optimally locally adapted. But when we look at genomic data, what kinds of clines do we expect to see? And that's really going to affect how the underlying drivers, if you will, of maladaptation, and also indicate how important regional variation is. And so picking up on work of Nick Barton's, we have some phenotypic line across a range. Do we expect to see parallel concordant clines in the genes that underlie that phenotypic trait, or do we expect to see localized genetic clines? And so Colin here focused in on SNPs that were associated with cold hardiness. So just a single trait, took a set of SNPs, so 186 SNPs here, and clustered those according to spatial covariance patterns along environmental gradients. So clustering SNPs into groups that showed similar patterns of variation across this landscape of lodgepole pine in Western Canada. And here are all these graphs. The clusters had anywhere from 7 to 25 SNPs in them. And I just want to contrast a couple of them with you. So if we look at this gray cluster 4, which had 25 SNPs in it, we don't see much variation among populations at cold temperatures. This x-axis is mean annual temperature. So it's relatively flat at very cold temperatures, but then we see a steep climb at warmer temperatures. And contrast that with cluster 6 here in purple, where we see a relatively steep climb at colder temperatures and then fairly flat at warmer temperatures. So we see that in different areas, in different portions of the climatic gradient, different clusters of SNPs are contributing the variation to these populations. And when we look at phenotypes, of course, we see quite a simple phenotypic climb across all of these. And if we look at this spatially, what we see is a big difference on either side of the Rocky Mountains. So the true northern Rocky Mountains, I know some people down there like to consider Montana the northern Rockies, but we don't quite see it that way. So the Rockies extend between Alberta and BC and then extend right up here. And what you see is this cluster 4 has a lot of variation in average frequency west of the Rockies, but not east. And cluster 6 has a lot of variation east of the Rockies in these very cold areas, but not west. Now, even though different clusters are contributing variation in different places, the good news out of this is that we saw almost every SNP had warm alleles present in almost every population. And cold alleles were present in almost every population. So the standing variation is very high, but the average frequencies differ a lot depending on where you are within the range. But let's get on to the maladaptation question, building on that, how we take information like that and try to understand the extent of maladaptation of different populations. So Matthew Fitzpatrick and Stephen Keller introduced this idea of using gradient forests to model the turnover of alleles instead of species along climatic gradients, and to then use those models to project maladaptation to future conditions. And very nice recent review by Capblanc et al. Using these kinds of methods. So we've been applying these to different data sets from our genomic studies. So first of all, here is some work that was done by UAU, who just finished her master's degree here, working with Tongli Wang, and she used the SNP data set that I've been talking about from lodgepole pine, from the Adapt Tree project. And she looked at using a full SNP set, or just using candidate SNPs from genotype environment associations and GWAS to project maladaptation of lodgepole pine populations under 2 climate change scenarios, RCP 4.5, which is a moderate level of climate change, and RCP 8.5. which is a more severe, of course, level of climate change. And so we see somewhat different patterns here, depending on if she used the full SNP set or the candidate SNP set. And I would expect the candidate SNP set to give us a better picture of that future level of maladaptation of populations. Now, she hasn't yet done the comparison with phenotypes to validate this approach. But I do want to point out with the full SNP set, if we see these areas projected to have higher levels of genetic offset in the south and along the sort of sub-maritime zone, just inland from the coast. And what's interesting about this is these are areas that if you take a climatic niche model for lodgepole pine, you expect to see lodgepole pine dropping out of at least these southern areas. So it's not clear to me yet whether we're getting a signal that these populations are experiencing greater maladaptation, or whether what we're seeing is the areas that are moving outside of the species climatic niche and tolerances. But it's an interesting area to dig further into. Brandon Lind and Rafael Ribeiro are also to project maladaptation, but Brandon is using the common garden phenotypes from Raphael's experiment and specifically growth increment in the Vancouver environment to validate those projections. So he's saying, how far have populations been moved from their provenance to the Vancouver common garden environment, and how much maladaptation would be projected by gradient forests for that degree of movement. And here's the common garden experiments that were used. So Brandon, here's the common garden experiment in Vancouver, and Brandon looked at doing this for the coast variety only, the interior variety only, and the full variety. And this is very preliminary work, so I'm only gonna show you one graph from it. But so what's interesting is, first of all, if you look across both varieties, Brandon's relationship here, where the genetic offset, a lower, where he's got a pretty good relationship between the projected genetic offset and the growth increment. And so you might think that's a pretty good relationship, but if we look again at coastal and interior varieties separately, the relationship's much weaker. And so we're really taking a hard look at these data to say what should be the scope of populations included in an analysis like this to get reasonable data. Our common garden is very much in the coastal Douglas fir environment, and so we're probably not getting a good look at what will happen with interior Douglas fir phenotypes when we're so far outside of the varietal range. But one of the preliminary results is that gradient forest predicts offsets better within variety, better than climate or geography. So it's better than that, but stay tuned on that because there's a lot more work coming. So just to summarize, first of all, what we found is that local adaptation to climate in conifers is very complex and highly polygenic. And so we really need to look at patterns rather than specific genes, because we don't find big effect size. We find this very complex architecture of traits. For the species we're working with, those climatic drivers of local adaptation. And I would argue, if you can't get any other information, that this is sufficient to make some conservative translocations based on, because of climate change, depending on what species you're working with. But we really need to combine that with expert knowledge about climatic and ecological associations of species, factors that are important, things that we might not be picking up, non-climatic environmental factors. We're finding with these analyses, both phenotypic and genomic, that scale really matters. And we have to be careful, I think, about deriving projections across very large species ranges when perhaps we should be digging in a little more at a finer scale. And this is the opposite of what I would have said 10 years ago, because I'd say there are too many small scale studies and we need range-wide studies. And the final point here is that I think that we have to, with assisted gene flow, which I haven't specifically talked about much today, but we've really got to, on one hand, we've got to be cautious. We've got to be very cognizant of the risks of translocations, and they are very real. We've got ecological risks. We've got genetic risks like potential outbreeding, depression. But I think we have to weigh those risks against the magnitude of climate change effects under status quo management. And, we're seeing change already. And so we're going to have to balance that doing something against doing nothing. And it's tricky times. So with that, I have many people to thank. I've named quite a few of them already, but the whole Adaptree and co-Adaptree teams and all of the funding organizations, especially Genome Canada, as well as NSERC, and highlight there, who's a co-project leader on co-Adaptree and all the others involved, are co-authors on a new edition of a textbook that's coming out, and it's Conservation and the Genomics of Populations, led by Fred Allendorf. And well, it should have been out already, but hopefully it will be out early in the new year. And I think you can still get a discount if you're interested in ordering it early. Yeah, so you can get the link from Chris. Anyways, I welcome your questions, and I know we're up at 4 o'clock now, and some of you probably have to go, but I'm happy to answer any questions that you have, and I can keep an eye on the chat for those, or put your hands up.
Thank you for the excellent talk, Sally.
Oh, a whole bunch of questions. Chris put up his hand and he took it down again. I'm just going to go by what I see on my screen. Or do you want to handle this, Miranda? How do you feel?
Yes, I am happy to handle this. Okay. I'm going with what I see first. So Ethan, you have a question?
Yeah, Sally, that was a really great talk. Thank you for providing it. So one of the things that became abundantly clear to me throughout it is just the lack of data we have for species in areas that typically are not big timber species. Mm-hmm. And so I'm just sort of wondering what your ideas are or recommendations are for when you don't have that level of intimate knowledge of these species and their associations. Is it just, making your best inference towards potentially looking at assisted migration? Or would you really recommend trying to find the capital to set up those sort of longer term experiments to get some of that knowledge, to make up some of that deficit there?
So we don't have time for longer term experiments. That's the sad truth. If we're talking about trees, if we're talking about tree species, we did an analysis that was published in 2016 where we looked across all the species that had common garden studies, tree species in Western North America, temperate species. So not into super dry areas, but it would have covered many of the trees you've got in Colorado, at least in mid and higher elevations. And we found remarkable consistency of the climatic factors that were and sort of relationships with growth. And so I would say that you could take the information that's available now on climate variables that are important and start, and if you're planting trees. I mean, that's a big if. But if you are planting trees for restoration or for reforestation, then going to a slightly warmer environment to make up for climate change that's already happened is, to me, something that you better to go ahead with it than to wait to put a long-term trial in the ground. There are lots of older provenance trials that will tell you something about what those climatic drivers are. If you have money, do a genotype environment association test and see if you see what climate variables pop up. But I'll bet you it will, if you were to match temperature, mean annual temperature, coldest month temperature, and summer precipitation. And you can use a tool called Seedware that's been developed by Brad St. Clair out of the US Forest Service. That would give you a pretty good guess. Is it going to be perfect? No. But is it going to be better than doing nothing? I think it is. So I hope that helps.
All right, thank you. Jeffrey Morris.
Yeah, thanks, Sally. So as you know from our previous discussions, I don't work on forests at all, but moving here to Colorado a year ago and exploring the forest, I've been thinking a lot about these questions about drought and climate change. And one hypothesis that's been like I've been wondering about, that I'd be interested in your thoughts on, is the role of like overcrowding, if that, I don't know if that would be the right term, but in the crop world, people like to decompose the environment part of GDPE into the environment, what we call the environment, which is the part you can't control, and management, the part you can control. And I've been just wondering how much the management practices like fire suppression, or like all the trees is growing up after being logged in the same kind of cohort, how much that kind of changes which alleles, if at all, are drought adaptive or not, compared to the historic, management practices. Any thoughts?
Yeah, I think that's a really good question, and we don't have a lot of good data on that. There's a very good study on the coast that looks at that with coastal Douglas fir, and generally found that the trees that were doing well at wide spacing were doing well at narrow spacing, but these are not very dry environments. People are thinking a lot about tree spacing and even planting with respect to fire risk, and whether or not and drought, but also just kind of what tree spacings are associated with what risks. The analyses I've seen coming out of our big fire years, which were 2017, 2018, and 2021, suggests that if you've got really bad fire conditions, it doesn't matter. Everything burns, but it may make a difference, and certainly having that less dense stands makes sense from a fire standpoint, and should provide more moisture. In our driest ecosystems, I think that our driest forested ecosystems, I think that's going to be really important, as is partial cutting systems that keep shade in the mix. But we've got pretty much one forested zone that's absolutely critical in. But we're going to have to do more to moderate those environments, more partial cutting, more alternative silvicultural systems.
Okay, fascinating. Thank you.
All right, Ruth.
Hi, Ruth.
Ruth, you're on mute.
There I go. There's two of me. Is it echoing?
Yes, but I can hear you okay.
Okay, I'm so sorry. Oh, that's painful. Let's see. Does that work? It's no better.
You might want
I'm
to
going
type
to
your
type my
question.
question.
Yeah.
Ruth, the other option is I'm just wondering if if you just go off of one computer and then the volume's off on any other computers that are linked in, that might prevent the echo. Well, okay. Well, while we're waiting for Ruth's question, I have a question of my own. So I'm just a little curious. You mentioned when we met earlier that UBC has been planting trees, or at least some forester managers have been planting trees recently that using kind of a more climate adapted framework with planting specific genotypes from warmer areas. And I'm curious of what's been observed so far. Is there much greater survival among those individuals or among those plantings where it is generally planted with those more drought adapted genotypes, or is there a lot of kind of frost damage mortality occurring?
So I think it's too early to really assess that. So we have a policy. So in BC, 95% of the forests are on public land, and there is policy that dictates what you can plant for reforestation. So the companies that have the logging rights have to reforest. They have to plant and ensure that those trees get to a certain size and certain number of trees before their responsibility is done. So they're quite keen on getting that done quickly. The seed source policy now has shifted from a seed zone approach where you source seeds within to a climate-based seed transfer system where movements are from warmer to cooler sites. But it's an incremental shifting policy that will change over time. And right now that new policy is optional. Foresters can use the old zone-based or the new climate-based seed transfer. And something like 60% of the trees being planted within a year of that new policy had shifted to the climate-based seed transfer system. It does provide them with some more flexibility, but anyways, it's based on long-term provenance trials, and as well our genomic analysis developed that's part of that system. So there's lots of long-term studies that have tested movements substituting space for time. But I think it'll be a few more years before we can really take a good look at how that's affected things like survival and growth. And I know last year and this year with droughts, we've had a lot of drought-related mortality, and I think that has happened. It's just climate change and new range of events. Okay, here's Ruth's question. So I'll just read it. Really curious about the finding that cold tolerance comparison of genomic signatures showed 40 evaluating the genomic signatures of selection within varieties was really important. Yeah. Can you comment on that? Also wanted to say that I really appreciate the point of way aside the rescue for that last point. And I think that if we are going to migrate things, which I think we should, and trees are I think trees are a pretty easy sell on that because we have a lot of information about local adaptation. And they're so long-lived and just they're going to be so slow to adapt, although I think there's the variation there for that to happen. But it is interesting how little overlap we saw for the drought, in that GWAS. It's higher for cold between the two varieties, but but it is a surprising result. I'm very curious to see what comes out of the new analysis that's underway for about 8 species. So we've got the lodgepole pine, the interior spruce, but we're adding Douglas fir, western larch, jack pine, Norway spruce, Scots pine, and I think there's one other. And it will be very interesting to see the overlap there. I think that result with drought just reflects, I'm not, we're trying to validate those markers for drought right now. Maybe that's where I should start. And so that GWAS, it was this case control approach. We didn't have a lot of statistical power with that. I'm very curious if those SNPs that are candidates from that drought experiment really hold up in the validation experiment that Judith's doing right now, because I feel like we don't have a handle on drought. For cold, I'm not surprised we see more overlap. We also see things that are involved in cold responses in Arabidopsis popping up when we do cold related GWAS. So just a more consistent genetic basis and fewer alternative mechanisms, perhaps. I don't think that was a very satisfying answer, but stay tuned.